In this video, we'll talk about cell division, that's mitosis and meiosis. We begin talking about the cell cycle for eukaryotic cells. Basically, we're talking about eukaryotic cells. Prokaryotes, that's bacteria cells, don't undergo this kind of cell division. Basically, they don't. Bacterial cells undergo binary fission. That's how we call the cell division there, binary fission. So we're dealing on eukaryotic cells here. Now, eukaryotic cell cycle. A cell, the general life cycle of the cell is it grows, it lives and produces children, or produces daughter cells, how we call it in biological terms, daughter cells. And so a cell has to grow, and this period of growth is called interphase. It's called interphase. While this process, this very short M phase here, you can see here, M signifies mitosis, is the reproduction phase. It produces daughter cells. So as we said, interphase, which is the majority, and mitosis. Interphase, mitosis, cell cycle. I think that's pretty basic. Okay, we start with interphase. Interphase, we divide into three phases. I'm sure we noticed that in the diagram. Three phases that comprise interphase. Interphase has the first phase of interphase immediately after the cell divides, forms. That's at the beginning of the life of a new cell. It's the G1 phase where the cell just grows and lives its normal life. Grows, eats, sends out waste products, and does whatever I want to do, basically play around, and yeah. Then the next phase after G1 is synthesis, the S phase. We call it the S phase too. From this diagram you can see. I don't know it's very clear, but S phase anyways. Where the DNA multiplies. A cell, eukaryotic cells have a nucleus, and inside the nucleus we have DNA. Now it's important you take note, DNA during interphase, the DNA is very loose coiled. We see they are thread-like forms. Yes, that's very thread-like forms, and we call them chromatin here, chromatin fibers here. Very thread-like. I have difficulty saying that. Thread-like. Okay, so this DNA replicates. So DNA basically divides and forms new DNA. G2 phase, after S phase, the cell is basically preparing for mitosis. So it's producing proteins that are concerned with mitosis, enzymes and energy, storing up a lot of energy for mitosis, because cell division takes a lot of energy. I mean, just like a woman in labor. Reproduction takes a lot of energy. Same thing with the cells. Okay, after interphase is the mitosis phase, which is this very short phase, but very important. Division of somatic cells, non-reproductive cells. Somatic cells are non-reproductive cells. All our body cells which are not reproductive. That is not the sperms, not the eggs, and yeah, all our body cells are somatic cells. Here in mitosis, a single cell divides to form two daughter cells, and the daughter cells are identical to the parent cell, completely identical, except you have a problem. There is a mutation or something that creates a problem, but ideally daughter cells are identical to parent cells, same number of chromosomes, everything. So remember we said cell DNA exists in the form of chromatin, which are loosely coiled threads during the interphase. Yes, because basically it's useful, contains the information needed to produce proteins and to control normal activities of the cell. It has to be uncoiled, easily accessible. Meanwhile, mitosis, you don't need to access. Everything for mitosis has already been prepared. So the DNA has to coil and become very condensed, as this brother we can see here. This guy is condensed, and take note, we call this guy a chromosome. This guy here, this is condensed DNA. Yes, you can see he's telling his dude, I can't believe you're not your condensed DNA. Mitosis is in 5 minutes. Yeah. So he has condensed, and we can see here he's kind of a double, yeah, 2, and that's because DNA replicated. Remember we said during the S phase there is replication of DNA, so it replicates, and this is attached at the center here. Yes, attached at the center here. And these 2 chromatids, we call them chromatids here, sister chromatids anyways. Yes, we call them sister chromatids because they are coming from the same parent DNA. So DNA forms chromosomes, and these chromosomes are made of 2 sister chromatids during mitosis. Okay, mitosis is divided into 4 phases: prophase, metaphase, anaphase, and telophase. And can I say a little PMAT? Yeah, it just came to me right now. PMAT, we can say PMAT: prophase, metaphase, M, A, anaphase, and T, telophase. PMAT. Then followed by now these phases is division of the nucleus, of the nuclear material. So the nuclear material divides into 2, separates into 2. Then now cytokinesis is division of the cytoplasm. The cytoplasm of the cell has to divide so that it finally forms 2 daughter cells here. As you can see, this is the cytokinesis here. You follow the red laser on the screen. Cytokinesis, where this cell divides into form 2 daughter cells finally. Okay, we begin with prophase. Prophase begins with condensation of chromosomes. Yes, so chromosomes condense during prophase, and spindle fibers form. Spindle fibers are formed by these centrioles. Centrioles are organelles that are generally around the nucleus. And centrioles are very important, spindle fibers. The spindle fibers are the ones aligning the chromosomes. So they are the one kind of attaching the chromosomes to, because remember, chromosomes are free, we said. Now, during prophase, there is disintegration of the nucleus. So your chromosomes are basically free in the cytoplasm. So these centrioles produce the spindle fibers, which are the ones that hold them in order. Yes, they are kind of a skeleton, a cell skeleton, microtubules. Spindle fibers are made of proteins called microtubules. And these centrioles are very important for mitosis, yes. Some cells in the body do not divide, cannot divide, like our neurons. Our neurons cannot divide. Our heart cells cannot divide. Yes, the muscle cells of the heart cannot divide. Basically, even skeletal muscles cannot divide. And it's funny, but because they don't have centrioles. Yes, basically. So neurons do not have centrioles. So they can't divide because they cannot form microtubules. So no centrioles, no microtubules, no cell division. It's as basic as that. So prophase, formation of microtubules of spindle fibers, condensation of chromosomes, and disappearance of the nuclear membrane. Metaphase, our chromosomes align at the equator, at the center of the cell. You can see here, they are aligning, and that's because of our microtubules our microtubules that attach to the kinetochores, the center of the chromosome. They attach it, they hold it firm. During anaphase, there is our sister chromatids finally separate and move to the poles, each pole. You can see they're pulled by the spindle fibers to each pole, and they go to each pole. That's anaphase. And during telophase, finally, which is completely opposite of prophase, telophase, complete opposite of prophase, there is formation of the nuclear membrane, and the DNA, the chromosomes, decondense. So they go back to that thread-like form, that chromatin form that was in interphase during telophase, actually. Yes. And then the last step will be cytokinesis, where the cytoplasm divides. Now, the difference between plant and animal cells is that, remember, plant cells have cell walls, cellular cell walls. So in cytokinesis in plant cells, it's just formation of a cell plate, of a new plate at the center. While animal cells, the cytoplasm has to actually pinch off and divide. Okay, we move on to the next. In the body, we have somatic cells and sex cells. And the sex cells are the gametes. Now we have another kind of division in cells, in eukaryotic cells, called meiosis, where instead of A cell dividing into two, like in mitosis, two daughter cells, it divides into 4 daughter cells, and the 4 daughter cells are not identical. They're different from each other, different genetic material from each other. Now, generally, it said diploid cells are cells in our body. Now, humans, I would say example of humans directly. Humans have 46 chromosomes, and those 46 chromosomes actually exist in pairs. Our body cells exist in pairs. So we say we have 23 pairs of chromosomes because each chromosome has a homologue, a kind of identical sister. Yes, so our chromosomes exist in pairs. So we say our somatic cells are diploid. Yes, and we have 2 sets of chromosomes. Each chromosome has its homologue. It's kind of partner in crime, or partner for short, yeah. And we have other cells which are the gametes or the sex cells, the sperms, the eggs, which are haploid, N, designated as N. Now, yeah, it's just an explanation. And actually, what determines the sex of humans is we have 23 pairs of chromosomes. So we have 22 autosomes. Yes, 22 pairs of autosomes, 22 pairs of autosomes and 1 pair of sex cells. Autosomes are the normal body cells, and the sex chromosomes is the 23rd pair. Now, what differentiates male from female is this 23rd pair of chromosomes. In females, we call them XX. We have 2 X chromosomes for the 23rd pair. And for males, we have 1 X and 1 Y. So, a karyotype is just the genetic representation of a cell, the complete genetic representation of a cell. So, question here is how many homologous pairs are in each karyotype? How many homologous pairs? We can see here we have each. This is a pair is what I said. Chromosomes exist in pair, kind of 1 pair is identical. Yeah, so we have 23 pairs in human cells, and this is the sex pair, X and Y, means this is a male. How is the bottom karyotype different from the top 2? This is haploid. It doesn't exist in pairs, so it's haploid, while this is diploid because they exist in pairs. So I hope that's clear. So during meiosis is basically sexual reproduction. Anything that undergoes sexual reproduction should have meiosis. Now, we said our body cells are 2N. It means when sexual reproduction is there to produce a new zygote, because basically during sexual reproduction there is in humans there is a sperm cell which meets an egg, and both of them fuse to form a single zygote. Now that zygote has to be 2N. It means that the 2 parent, the sperm and the egg, have to be N and N, or haploid, so that we don't have a production of a zygote that is 4N, for example. Yes, so our sperm and eggs have to be each N. That's why meiosis is important in sex cells. We reduce. By the way, we also call meiosis reduction division. Yeah, another name for meiosis is reduction division. Okay, so yeah, sexual reproduction in humans. We form haploids, gametes, and which fuse to form the zygote.